हिजो अस्ति आदि पढ्दै I had to leave है त्यो सेकिङ इन्क्युबेटर आएको थियो कि अनि त्यो सेकिङ इन्क्युबेटरलाई चाहिँ मिलाउनको लागि I had to leave the class. Okay, so in today's class we'll start from the beginning, and so we'll study about the different computational approaches to the biological problems. So there are certain problems in biology, and we can convert those problems into a computational one, and it will be easier if we do. It will be easier if we do it so. We'll see किन यस्तो हुन्छ भनेर अब biological problem like computational problem मा किन लिएर जानुपर्छ भन्ने what is the need भन्ने कुरा we'll also identify. So the first is that using public databases and data formats. So one of the first skill of biotechnologist, biologist is to learn the online search tools to find information, because most of the journals these days they publish their weekly or fortnightly or monthly subscription in the online format. So in the past, what the students they had to do is that they had to physically go to the library and start searching for the literature in the different indexes. But now that's not the case. Now you can, with the help of Google Scholar or PubMed or Intrans, you can do the literature search in just a matter of time. You can have like hundreds and thousands of journal papers relating to your field of inquiry. So you can find links to the most scientific publication you need online. So there are central databases that collect reference information so that you can search dozens of journals at once. So even for this, you need to have a certain computer knowledge, and behind the back there has to be this technology, there has to be this development in hardware and software by the virtue of which all these scientific papers, this journal, these research findings, they can be published online. There is a whole software and hardware business. एकदमै डिटेलमा छ र त्यो गर्ने त छँदैछ at the back र front end मा चाहिँ कसरी सर्च गर्ने भन्नेको लागि पनि it's a computational problem. त्यो भएको कारणले it becomes easy if you search through online. Even that's why you need computers. That's why you need the power of computers. And then second is the problem of sequence alignment. The sequence alignment and sequence searching. So sequence alignment, why we do sequence alignment? Because with the help of sequence alignment, we can deduce homology. Right? We can deduce homology. Sequence same छ भने के हुन्छ, right? Homology. दुईटाको sequence same छ भने, so यसको sequence as you can see local alignment मा यसको same छ sequence. So what does that mean? Maybe यसको function, माथिको function, if you know the function of the upper region of this segment, then you can deduce the function of the lower segment as well, right? So this is the power of sequence alignment. So being able to compare the DNA or the proteins sequences of complete and impartial matches is one of the fundamental things that you will do in biology. So why do we do the sequence alignment and sequence searching? I said to identify homology, to identify certain structural motif, right? Structural and functional motif, right? So if this was the case with a protein, right? The earlier case, this was the case of protein. Then what we can deduce is that if they have a similar kind of sequence, then what you can mean is that the residue are similar, and maybe they fold in a similar way, or they have the similar kind of function. So it may be due to homology, or it may be due to an independently convergent evolution. So there is this concept of convergent evolution. Convergent evolution. So what do you mean by convergent evolution? Anybody, do you have any idea about convergent evolution? So what happens in convergent evolution is that two distinct lineages, they can develop similar function independently, right? So even so there is a, this हाम्रो retina there is this light sensitive proteins and even in bacteria you can find these light sensitive protein. And if you do a homology search, sequence alignment, if you do the sequence alignment, then what you can see is that they share lots and lots of similar sequence similarity. You can observe them. And why is that? How is that sequence similarity? It's not due to homology, right? Homology भएको कारण भए त they should have the same ancestry हुनुपर्छ, original ancestral sequence same भएर चाहिँ निस्केको हुनुपर्छ, but that's not the case. It's because of due to convergent evolution. So by the help of convergent evolution also you can get the sequence similarity. So you what happens is that you can deduce function of the query sequence. So query sequence ko function pata launu sakincha. And what you have to realize is that so this target sequence is from database. So this is from database. So in database there can be many other target sequences. Maybe there are million target sequences, right? So if you want to do this activity, right, individually without the help of computers, then this will be a very difficult task, both the local alignment and the global alignment. So what do you do in the local alignment? You just search for the local regions, right? Just where there is maximum search. So you don't do for the whole, for the overall segment, right? You just do for where there is maximum amount of matches. And what you do in the global alignment, you search for everything, right, everywhere. So maybe so we are not aligning here. So we are not aligning here, right? So why we are not aligning here? Maybe there was a deletion event, right? There was a deletion event, right? Or maybe there was so maybe there was a deletion event, or sometimes there is a repetition. So there even can be some duplication event as well. So duplication event. So all these things, it will be very difficult for a human brain to scan. So there are millions of the target sequences, and so it will be very difficult to do these kinds of searches. So we'll study in great detail about the local alignment and the global alignment in our forthcoming lectures. So this is kind of sequence-based searching. It's a very key skill set for a biologist, because if we do this at the beginning, we can get lots and lots of information about that protein, right? So for example, if you want to find a protein and you have some idea about that protein, then what you can do is that you can search that protein in a database. So maybe it has an ATP binding site, right? ATP binding site. So if it's a maybe it is a transcription factor, and it has different kind of motif. So maybe it has a zinc finger motif or a helix turn helix motif. If you have this kind of information, then you can do the search in the database. There are different kinds of databases. We'll study about those kinds of databases in our forthcoming lectures. So there are the databases where you can find motifs and profiles, right? Fingerprints, regions. So all these kinds of databases you can search for protein, and that will be very helpful, and it will save lots of your valuable time. And thirdly, so identifying homologous sequence provides for a basis for phylogenetic analysis and sequence pattern recognition. So there might be certain information in here and that you can deduce. So sequence-based searching can be done online through web forms. So it requires no special computing skills, but to judge the quality of your search results, you need to understand how the underlying sequence alignment method works and go beyond simple alignment to other different kind of analysis. So since in actual case, so there will be different target sequences. So this is one target sequence, and this is another, right? This is another, this is third, this is fourth, and so on. And you can get different values for this, right? A different mathematical score. You can get different mathematical score. So on the basis of that score, you must come up with certain kind of inference. So whether this is a good score or not, whether on the basis of this score you can deduce homology or not. So all that kind of information, that's the post alignment task that you need to do. So these are one of the most important skills that you learn in this course. So another is the gene prediction. So there is one of the cluster of methods for attempting to detect meaningful signals in uncharacterized DNA sequence. So there are many sequences, and you need to find the meaningful regions. So in gene banks, we have many different sequences, and many of them are characterized, annotated, and many of them are not characterized or annotated. So on the basis of previous sequences, we can do some gene prediction. So, so what you can do is you can search for the promoter region, you can search for the terminator region, or you can search for introns and exons, all these kinds of regions. So you can search for the open reading frames, genes, and the exon splice sites, promoter binding sites, repeat sequences, and TNA genes that can help molecular biologists to make sense of the unmapped DNA. So this is a very important tool of the gene prediction that you can use. So मैले अस्ति पनि भनेको थिएँ क्या there are around assumption के छ भन्दा there are around 20,000 to 25,000 genes in human genome. This is a prediction, right? So as you can see, so there is a big range, right? 500, there is a big uncertainty. So why is that? The uncertainty is because of our gene prediction software, right? Different softwares. So if you input different parameters, they come with different results. So this number was considered to be around 40,000 in the beginning, and actually with the sophisticated gene prediction tools, this number is decreasing. So what you do is that there is the whole genome, right? Whole genome of the human, whole genome from any organisms, and we have identified some of the defining characteristics of the gene, right? So sequence properties of the genes. So we have many genes, right? We have many identified genes, annotated genes. So some of the sequences are characterized. So on the basis of these characterized genes, we can come up with certain softwares, right? Algorithm. So these algorithms, what they will do is that they will search for the new genes, and these softwares, so they can find new genes. So the algorithm will find new genes, and what we can do is that we can train the machine, so this will find more genes, and what you can do is that, so you can again feed that into the algorithm. So this way you can learn, you can teach your algorithm, and all this kind of things will come into this algorithms that can learn itself, in ML, machine learning algorithms. So on the basis of on this way, in this way you can predict more genes. So this is how you do the gene prediction part. So we will also study about this in greater detail in our forthcoming lectures, how we do the gene prediction and what are the different methods we can use in gene prediction. And another most important critical is the multiple alignment. So in earlier we studied about the pairwise alignment. This is the pairwise alignment. Here actually we are just pairing two sequences at a time. This is a pairwise. So the query sequence may be many. The target sequence, it can be millions of target sequences, but what you are doing is you are comparing one at a time. Right? This is a pairwise alignment. Every time you are making one compare, so the target sequence and query sequence. So this is a pairwise alignment. Global alignment is also a pairwise alignment, but in SA, or the multiple sequence alignment, you are doing many rounds, many sequence alignment at the same time. So this is one sequence, this is two sequence, this is third sequence, and so on. So what is the benefit of doing a multiple sequence alignment. So the benefit of doing multiple sequence alignment is that you can discover certain reasons. So maybe you can identify. So if you know, so if you have characterized the first three protein, okay, this is something you have already done. These are characterized, right? This is characterized, and you know that this region. So this region is a helix region, and then you know this is a DNA binding region. And what you can infer is that even the rest of the segments, even these segments, you can confer that these are also your these are also your DNA binding regions, or your helix regions, or seat regions. And these are here you can also identify key residues, right? Some conserved residues, conserved residues. So this is a very important uh, thing that we need to do in biology. So multiple sequence alignment, and as you can see, multiple sequence alignment is a very difficult problem. Computationally, it is also very difficult. So in multiple sequence alignment, what you do is that you make a pairwise alignment of many sequences at the same time, and from this you can identify sequence homology, as well as you can identify the gene family, right? So you can identify domains, right? Maybe you can identify a certain. So last time I told you G protein coupled receptors, right? So, or you can find some tyrosine, tyrosine kinase family, right? Different kinds of protein, say tyrosine kinase family or exokinase family, right? All these kinds of gene family in proteins you can identify. So this is also about the, as you can see, this is also a visualization identification of the sites of the DNA or protein. Here you can so here you can visually identify certain regions. So these are certain key regions, conserved regions. So multiple alignment can also be quantitatively analyzed to extract information about the gene family. And multiple sequence alignment is also a very critical steps in phylogenetic analysis. So before conducting any phylogenetic analysis, first step you do is the multiple sequence alignment. And on the basis of that, you derive these phylogenetic trees. So it's a very crucial. So we'll also study a lot in a great detail about different algorithms in multiple sequence alignment in our forthcoming lectures. So in phylogenetic analysis, what you can do is that this is the sequence-based phylogenetic analysis. And what you can do is that we have a sequence, right? Maybe this is a hemoglobin sequence, right, from different organisms. And what is our assumption here? That maybe there was this ancestral sequence, and this ancestral sequence, it it undergo mutation several times, and these mutation, they gave rise to species, right? And then we have this speciation events, and we have different these species, or this can be species or this can be individual as well. But since we are doing phylogenetic analysis, we'll just limit our talk to the species level. Right, the species level ma matra talk गरौँ है त. So this is the first species, and the other. So these two are the related ones, and these two are these and these and these and these. They are extremely far apart. So these are the ones with the greatest change, right? So since there is lots of sequence variation between these, right? So between these and these, right? Between these and these. So what we can conclude is that they were the one to have an speciation event in the beginning. So we can deduce this phylogeny on the basis of sequences. So a traditional phylogenetic tree or cladogram groups species into diagram that represents the relative evolutionary divergence. So branching of the tree that occurs farthest from the root separates individual species. Branching that occurs close to the root group species into kingdom, phyla, classes, families, genera, and so on. So these two are extremely related one, and the next is the related are these one, and these are related. So these two are related to these, right? And these are related to each other, right? And this is how these phylogenetic trees can be interpreted. The information in the molecular sequence alignment can be used to compute phylogenetic tree for a particular family of the gene sequences. So there are certain genes which can be used to do this kind of phylogenetic analysis. Not all genes they can be used to infer phylogenetic analysis. The branching in the phylogenetic tree represent evolutionary distance based on sequence similarity. Scores or the information theoretic modeling of the number of mutation steps required to change one sequence to another. So we'll also study about different phylogenetic methods in our forthcoming lectures. Or different methods are right? There are character-based methods or character-based methods or distance-based methods or त्यसमा पनि different किसिमको distance based मा पनि there are different algorithms like the maximum parsimony भन्ने हुन्छ, right? Maximum likelihood भन्ने हुन्छ, different methods हरू हुन्छ. Even on the basis of those algorithms we will come up with different phylogenetic trees. So यो phylogenetic tree बनाउन पनि it is very difficult है त. So यसलाई पनि computationally यसलाई चाहिँ algorithm generate गरेर you can generate algorithm generate गरेर you can come up with these phylogenetic trees. त्यसको पनि statistical significance हुन्छ, which tree is more likely, which tree has the maximum likelihood भनेर all that thing we'll study in our phylogenetic analysis chapter. So अर्को भनेको के छ भन्दाखेरि त्यो कन्सेन्सस सिक्वेन्सेसहरु प्याटर्न्सहरु पत्ता लगाउन प्रोटिन्सहरुमा कुनै प्याटर्न छ भने we want to identify. So this kind of pattern we can identify, right? So कुनै प्रोटिनमा maybe this is an ATP GTP binding site छ भने we can identify. So यसको consensus pattern हो AZ, right? अनि it can be so there's a special way to read this है त. यो read गर्ने चाहिँ there is a special way, right? And usually this is a consensus pattern or is to DNA sequence motif pattern even these we can identify. So with the help of these sequences uncharacterized sequences with the help of this kind of pattern we can search for these sequences in uncharacterized DNA. So I give you a uncharacterized DNA and I want you to find different motifs or different structural elements, then how different functional element, different domains, and how will you do that? So what do you do that? There are certain databases, right? The databases are right, and with the prosite like the common prosite database, right? Prosite database, and from that prosite database, what you can do is you can submit your uncharacterized protein sequence, and from that uncharacterized protein sequence, that database will give you the final result. If this is your uncharacterized DNA, and that will do the searching, and you would say maybe this is an ATP binding site, maybe this is another helix, right? This is an helix. So this is another domain. So this is a beta seed domain. These are beta seed domains, right? So all this kind of information the database can give to you. So a motif is a sequence of amino acid that defines a substructure of a protein that can be connected to a function or a structural stability. So what you can do is that, so even this is this sequence and so in a group of evolutionary related gene sequences, so motif appears as a conserved site. So it is very difficult. What happens is that you have to understand, right? So motif they tend to get preserved. About motif, so suppose this is a sequence, right? This is a protein sequence. So if you take domain, if this is an alpha helix domain, if this is an alpha helix domain, then your domain your motif there will be less chance, right, of mutation occurring. If mutation occur, there will be selection pressure. So what happens is that your evolution while the evolution is working its way, so your region एकदमै selection बढी हुन्छ. Here, if the mutation occurs, then those, because since it will destroy the helix, right? If mutation occurs हुन्छ यहाँनेरि, if mutation occurs, then it will destroy the helix. It will tend to destroy the helix. It will destroy the helix. It will destroy the helix, and if what happens if it will destroy the helix, then the property of the enzyme or protein will be changed, right? The property of the enzyme will be changed. So what happens is that then there will be a selection pressure. So normally what happens is that अब यसमा change हुने बित्तिकै maybe there is a bad change भयो भने, so this kind of individuals they will be removed from the population. So what we see that, so these kinds of motif or this kind of domains, they tend to be conserved. So अरू साइट्सहरुमा चाहिँ there will be the appearance of mutations. There will be the नयाँ sequencesहरुमा चाहिँ there is this chance of new residues occupying those spaces. But used to conserved sites, this is less likely. So we can search, right? We can search for those sequences. So non-essential part of the gene sequence will diverge from each other in the course of evolution. So the conserved motifs regions show up as a signal in a sea of mutational noise. So other regions there won't be as much mutational rate. So in your region so you say motif को region domain को region protein को domain को region there is the less chance of mutation. So you can identify these regions in a sea of mutational noise. So sequence profiles are statistical description of the motif signals. Profile can help identify distinctly related proteins by picking out motif signal even in a sequence that are diverged radically from the members of the same family. So what you can do is you can generate these kind of profiles. This is a profile. So what happens is that there is the chance, right? So not every like sequence मा पनि motif मा पनि एउटै motif हुन्छ भन्ने छैन. That there is this chance. So यहाँ हामी के देख्छौँ भन्दाखेरि GAA हुने chances चाहिँ it is maximum, right, in these regions. So there is frequently occurring चाहिँ के हुन्छ GT हरु चाहिँ they occur frequently. And what we see that, so यो रिजनमा अरु A occur हुने chance र यो पोजिसनमा T occur हुने chance there is quite low. So we can construct these kinds of sequence profile and we can do search. You can do search on the basis of the sequence profile. So this is another example of a sequence profile. You
right side or the right side consensus pattern मा हैन किन त्यो ब्र्याकेटहरु लेखेको छ?
Okay, the bracket को meaning के हो भन्दाखेरि what is that? So at the first site यो A पनि हुन सक्छ G पनि हुन सक्छ. It can be A or a G. So यो X4 भनेको चाहिँ here it can be any amino acid. यहाँ चाहिँ any amino acid हुन सक्छ, right? And चारवटामा चाहिँ सबै चारवटामा. So here is the A or G हुन सक्छ. सुरुमा A हुन सक्छ, त्यसपछि or A or G हुन सक्छ, right? अनि त्यसपछि के छ भन्दाखेरि and second, third, fourth र fifth मा चाहिँ it can be any amino acid, but यो पोजिसन मा चाहिँ it has to be G र यो पोजिसन मा it has to be K र यो पोजिसन मा चाहिँ यहाँ S पनि हुनु हुँदैन T पनि हुनु हुँदैन. So this is the consensus pattern. यसलाई हामी consensus pattern भन्छौँ. यसको चाहिँ यो चाहिँ एउटा चाहिँ nomenclature छ, how you write it, right? इसको बारेमा पनि we'll study while we'll study प्रोजाइट पढ्दाखेरि we'll study प्रोजाइट मा चाहिँ कसरी सर्च गर्ने भनेर. So thus this is how अब यसमा चाहिँ what you can do is that यसमा 50-50 हुन सक्छ. A and G can be 50-50, right? So यसमा X मा it can be any protein residues, right? Any residues, चारवटा. So यहाँ it can be A or it can be G. And this पछि चारवटा 1 2 3 4, then it has to be G, then it has to be K, then anything except S and T, right? So this is how consensus sequence are made. and यो चाहिँ यसको डायग्रामेटिक रिप्रेजेन्टेसन हो. सो as you can see,
यस र टी बाहेक अरू भन्नुभयो, कसो थाहा भन्नुभयो त्यो माइनस लेखेकोले सबैमा चाहिँ
नो
माइनस
नो नो ए ओके सरी सरी यसमा चाहिँ के हुन्छ नि सरी यस यस अर टी है त यहाँ यस अर टी यहाँ जुन चाहिँ त्यही त हो नि राइट कि यस अर टी यहाँ जुन चाहिँ हुनु पर्यो कि यस हुनु पर्यो कि टी हुनु पर्यो कि यस हुनु पर्यो कि टी हुनु पर्यो यहाँ यस हुनु पर्यो कि टी हुनु पर्यो सिमिलर लाइक इन द एआरजी यहाँ यो जस्तै त हो समटाइम्स यु हेभ यस्तो स्क्वायर ब्राकेट पनि हुनसक्छ यो स्क्वायर ब्राकेटको चाहिँ यस र टी भएको भए चाहिँ एभ्रिथिङ एक्सेप्ट एस एन्ड टी भन्छ क्या यो स्क्वायर ब्राकेट भनेको चाहिँ स्क्वायर ब्राकेट भनेको चाहिँ ए अर जी भनेको यहाँनिर तिनवटा भएको भए यहाँ ए जी र के भएको भए ए अर जी अर के भन्ने मिनिङ हुन्थ्यो राइट सो दिस इज देयर इज दिस नोमेनक्लेचर है त यो यु क्यान डु यो रेगुलर एक्सप्रेसन भन्ने यु क्यान डु द सर्च है त रेगुलर एक्सप्रेसन भन्छ रेगुलर एक्सप्रेसन्सहरू भन्छ प्रोग्रामिङ ल्याङ्ग्वेजमा पनि एकदमै युज हुन्छ है त यो धेरै ठाउँमा युज हुन्छ रेगुलर एक्सप्रेसन राइट बट विल स्टडी अबाउट दिस प्रोसाइट सेकेन्डरी डेटाबेसेसहरू पढ्दाखेरि विल स्टडी अनि सिमिलर यसलाई यसरी पनि डायग्रामेटिकल्ली पनि वी क्यान रिप्रेजेन्ट सो दिस इज हाउ वी क्यान एक्सट्र्याक्ट इन्फर्मेसन फ्रम एन अनक्यारेक्टराइज डिएनए मैले भन्न खोजेको के हो भन्दाखेरि इभन दिस यु क्यान यो पनि क्वेरी हुनसक्छ दिस क्यान बी क्वेरी against the database, right? There's a big database, and now this can act as a query. So even this is the pictorial representation, right? So computational representation पनि हुन्छ कम्प्युटरले जेनेरेट गरेको how do you generate this, right? So computational representation पनि हुन्छ. So that computational representation like what you can do is that you can this can be as a query and you can send that query to the database and the database ma चाहिँ मेरो uncharacterized DNA मा चाहिँ I can find out whether कहाँनेरि छ भनेर लेख्स ए binding protein. So if I suspect that my DNA is my DNA sequence चाहिँ एउटा चाहिँ लेख्स ए binding protein को DNA sequence हो कि होइन भनेर चाहिँ if I want to do it यो DNA sequence मा चाहिँ लेक्स ए भन्ने प्रोटिन चाहिँ बाइन्ड गर्ने if this is my sequence DNA sequence यो DNA sequence मा I suspect that लेक्स ए प्रोटिन can bind and यो लेक्स ए प्रोटिन रिप्रेसर प्रोटिन यहाँ बाइन्ड हुन्छ कि भन्ने if I think if I hypothesize what I can do is that यो sequence यो चाहिँ I can do the search यसको यो यसलाई चाहिँ यो sequenceलाई यो queryलाई चाहिँ I can do search and then I can identify same हुँदैन कि exact same हुँदैन कहिले पनि because that's why त्यही त हो नि there is noise मा signal छ है त so we want to identify that signal सधैँ एउटै हुँदैन right because there is this variation जहाँ पनि हुन्छ right even at the so मोटिभहरुमा पनि variation हुन्छ I hope you are clear है त अब any problem मा कसैलाई problem छ
छैन
okay this is the another holy grail है त एकदमै holy grail भन्छ यसलाई एकदमै holy grail problem भन्छ so this is about the predicting the protein structure on the basis of the sequence so it's very difficult to determine the structure of protein the extracrystalline first you need to purify it and then you have to crystallize it and then you have to do X-ray crystallography and all that thing. We have already studied all about that, right? So now it would be extremely easy if we could do some structural prediction just on the basis of sequence, right? So is it possible or not? Is it possible छ कि छैन? Is it possible?
So
right so if it's possible let's do it because we studied about Anfinson's experiment and what was the conclusion of Anfinson's experiment that to the native structures are confirm to final confirmation cha right biological activity highest biological activity wala confirmation cha त्यसको information that is encoded in the protein sequence itself, right? There is, like, maybe अरू कुरामा चाहिँ maybe there are sometimes there are chaperone molecules, chaperone protein molecules that will aid in uh, that will aid the protein to fold in the correct way. But there are many proteins that they will fold automatically in that medium, in that solution. So now what we want to do is that on the basis of this sequence we want to construct the model. So this is the model. I want you to be extremely be cautious. So there is difference between a model and reality. So what we can do is that on the basis of certain assumptions, on uh, certain understanding of our reality. So we have certain understanding of that reality, and on the basis of that, what we can do is that, so we can do all these things and we can come up with our final answer. And final answer is our model. So हामीसँग के छ भन्दाखेरि we have lots and lots of data, right? We have lots and lots of data. So we know that if there is certain sequence, right? हामीले धेरैवटा चाहिँ प्रोटिन्सको हामीसँग सिक्वेन्स पनि छ, and we have its corresponding 3D structure as well. So do we हामीसँग यो छ कि छैन? We have sequence and its corresponding 3D structure. छ कि छैन यो इन्फर्मेसन हामीसँग? Do we have this information or not? So डेटाबेसमा हामीसँग धेरै छ, यस्तो सिक्वेन्स पनि छ, त्यसको 3D स्ट्रक्चर पनि छ. And on the basis of that, so यो हाम्रो के भयो भन्दाखेरि, so हाम्रो यो चाहिँ के भयो भन्दाखेरि, this is our training set भन्छ क्या यसलाई, training set भयो. So we will train our algorithm on the basis of this. हैन, यस्तो यस्तो किसिमको सिक्वेन्सेसहरु छ भने this will fold in this way. If this is the kind of sequence, then it will fold into certain domains. So on the basis of that, what we can do is that sequence, and we will do the secondary structure element prediction, and then we will do a further Monte Carlo simulation, and then we'll come up with other random ways this can arrange, and we can do the clustering, and this we can do some certain loop construction and then clustering, and then we'll do molecular dynamics, and then we'll do some refinement. Because what you have to understand is that the protein molecules, they are not stationary. Actually, these protein molecules, they are actually in a dynamic state. They move around. There is lots of movement of these molecules, and we have to do certain molecular dynamics refinement as well, and then you come up with the final model. So here you can use the bioinformatics and computational tools to go from the sequence to the final model. So methods such as secondary structure prediction, threading can help determine how a protein might fold, classify it with the other proteins that have similar topology, but they don't have to have a detailed structural model. So you don't, first manage, you don't come up with a detailed structural model. So in the first, you come up with certain, and then what you do is that you refine that model continuously.
So to trading भनेको के हो?
सो यसमा चाहिँ के हुन्छ भन्दाखेरि रिपिटेडली वाट यु डु इज द्याट यसमा चाहिँ कन्टिन्युसली त्यही स्टेप्सहरू कन्टिन्यू गरिन्छ, अनि यु विल सी कसरी चाहिँ यो प्रोटिन फोल्ड हुन्छ भनेर हेरिन्छ है त. यो सबै हामी चाहिँ यो होमोलोजी मोडलिङ भन्छ पछि है त. यो होमोलोजी मोडलिङ भन्छ. यो होमोलोजी मोडलिङ पढ्दाखेरि वी विल रिड इन डिटेल है त. यो प्रोटिन फोल्डिङको बारेमा हामी चाहिँ वी विल स्टडी इन डिटेल, एकदमै डिटेलमा पढ्छौँ. सो दिस इज हाउ यु बेसिक सिक्वेन्स चाहिँ के हुन्छ भन्दाखेरि चाहिँ सिक्वेन्स हुन्छ. सिक्वेन्सको चाहिँ फर्स्ट यु विल डु डिफ्रेन्ट यो मोडलिङ मेथड्सहरूबाट चाहिँ डिफ्रेन्ट स्ट्रक्चर्सहरू आइडेन्टिफाई गर्न चाहिँ वी विल ट्राई इट. सो यो चाहिँ होमोलोजी बेस्ड भएर गरिन्छ है त. होमोलोजी भनेको सो यु हेभ टु हेभ अ प्रायर अन्डरस्ट्यान्डिङ. ए प्रायोरी अन्डरस्ट्यान्डिङ चाहिँ हुनुपर्छ. ए प्रायोरी भनेको पहिले नै थाहा हुनुपर्छ है त. यो सिक्वेन्स छ भने यसरी फोल्ड हुन्छ भनेर ए प्रायोरी भन्छ, पहिले नै थाहा भएको हुनुपर्छ. सो यो चाहिँ होमोलोजी मोडलिङ भन्छ यसलाई. दिस इज समथिङ द्याट इज एक्सट्रिमली पपुलर. अरू मेथड्सहरू पनि छ है त. सो एब इनिसियो बेस्ड मेथड्सहरू छ. दिस इज एक्सट्रिमली कम्प्लिकेटेड. सो यसको बारेमा चाहिँ वी वन्ट बी डुइङ मच. यसको लागि चाहिँ सिन्स यसमा चाहिँ के हुन्छ, देयर आर लट्स एन्ड लट्स अफ lots and lots of variables or parameters धेरै भएको कारणले even the modern computational ले चाहिँ पनि यसलाई गर्न सकिदैन है त so this is more or less how we do the computer structure prediction है त in absence of homology there is no way to predict the complete 3D structure of the protein so still है त हामीसँग यो चाहिँ this is still our यो चाहिँ हामी अहिलेसम्म गर्न सक्दैनौँ if you don't have a homology then it will be very difficult to identify those protein sequences
सर योबाट हैन फाइनल मोडल आयो तर लास्टमा रियल मोडल कसरी निकाल्ने रियल
ए ओके द रियल चाहिँ के हुन्छ भन्दाखेरि रियलको लागि चाहिँ वाट वी निड टु डु इज द्याट यसको लागि एक्सरे क्रिस्टलोग्राफी नै गर्नुपर्छ सो यस्तो छ अब यस्तो हुन्छ यो हामी के गर्छौँ भन्दाखेरि सपोज मैले तपाईँलाई मोडलिङ गर्न दिएँ है यो प्रोटिन सिक्वेन्सको छ यो प्रोटिन सिक्वेन्सलाई चाहिँ आई टेल यु टु एप्लाई योर एल्गोरिदम्स and then come up with your model भनेर भन्छु right and then you give me a model है त यो एउटा चाहिँ मोडल you give me your model यस्तो किसिमको फोल्डिङ भएको छ भनेर र त्यो मोडललाई चेक गर्नको लागि चाहिँ अलवेज है त आई सुड ह्याभ यसको चाहिँ रियल स्ट्रक्चर राइट एक्सरे क्रिस्टलोग्राफीबाट आएको रियल स्ट्रक्चर हुनुपर्छ एन्ड वाट आई डु इज द्याट आई कम्पेयर इट सो कति स्योर छ भनेर सो आई डु इट मल्टिपल टाइम्स मसँग भएको यो चाहिँ आई हेभ दिस स्ट्रक्चर and I tell you make a model with your all your you say about you say secondary structure prediction गरेर threading गरेर right सबै गरेर चाहिँ you every time you come up with new नयाँ सिक्वेन्स दिए you will come up with new model यसको लागि चेक गर्नको लागि के छ भन्दाखेरि you have to have the real data रियल डेटासँग चाहिँ म्याच गरिरहेछ 99% म्याच गरिरहेछ राइट 97% म्याच गरिरहेछ दिस आर एक्सट्रिमली 90% भन्दा बढी म्याच हुने बित्तिकै दिस आर भेरी गुड म्याचेस सो एकदमै राम्रो म्याच दिइरहेछ भने देन वाट आई डु इज द्याट अब त्यसलाई चाहिँ युज गरेर आई कम अप विथ यो चाहिँ अब योबाट चाहिँ वी क्यान डु इट भनेर यो प्रुभ हुन्छ है त स्टिल द्याट विल बी भेरी द्याट विल बी एकदमै अनि त्यहाँबाट हामी चाहिँ विल डु सम प्रिडिक्सन the prediction गरिन्छ every time if those predictions they are met if they are fulfilled then what we assume that यो चाहिँ जुन टुल छ हामीसँग जुन सफ्टवेयर प्याकेज छ त्यो सफ्टवेयरले टुलले चाहिँ अल्गोरिदम्स छ द्याट इज वर्किङ भनेर सो स्टिल है त हामीसँग त्यही नै हो कुनै थ्योरी काम गर्छ कि गर्दैन भनेर चेक गर्नको लागि यु हेभ टु डु मेनी त्यो फल्साइबिलिटी भनेर हामीले पढाएको थियौँ नि फल्साइबिलिटी भनेर द्याट साइन्सको चाहिँ एउटा हाइपोथेसिस एउटा चाहिँ लले थ्योरीले चाहिँ द्याट सर्टेन एउटा पनि इन्स्ट्यान्स चाहिँ त्यो थ्योरीले प्रुभ गर्न सक्दैन भने त्यसलाई चाहिँ प्रेडिक्ट गर्न सक्दैन भने द्याट द थ्योरी इज रङ भने जस्तै हो स्टिल है त द्याट इज हाउ द मोडलिङ वर्क्स सो त्यसलाई चाहिँ अझ त्यो राइट छ कि छैन भनेर चाहिँ चेक गर्नको लागि यु सुड डु द एक्सपेरिमेन्ट्स either so if many times if it gives correct prediction then what you have to understand is that now you have to give it a status of a law or a theory that is how it works either so about other methods protein structure prediction make protein structure property analysis गर्न सकिन्छ so protein structures have many measurable properties that are interest to the crystallographer and the structural biologist. So protein structure validation tools are used by crystallographer to measure how well a structural model conforms to the structural rules extracted from the existing structure or chemical compound models, right? So this tool may also analyze the fitness of every amino acid in the structural model for its environment, flagging such oddities as blurred charges with no counter charges or large patches of hydrophobic amino acid found on a protein surface. So different tools we can do certain analysis. So these tools are useful for evaluating both experimentally and theoretical structure models. Different properties protein properties we can come up with these kinds of protein स्ट्रक्चर एनालिसिसहरू, जस्तै कहाँ के छ भनेर। सो डिफ्रेन्ट के छ, फिजियोलोजिकल केमिकल प्रोपर्टिजहरू के छ, इन्टरनल जियोमेट्रीहरू के छ, कहाँनिर चाहिँ क्याटालिटिक साइट्सहरू छ, यसको मेकानिजम के होला, केमिकल प्रोपर्टिजहरू के होला, कहाँनिर डिप क्लेफ्टहरू छ राइट प्रोटिनमा, राइट कहाँ डिप क्लेफ्टहरू छ, इन्टरनल क्याभिटिजहरू छ, वी क्यान आइडेन्टिफाई कहाँनिर चाहिँ यो कोफ्याक्टर बाइन्डिङ साइट्सहरू छ, एक्टिभ साइट्सहरू छ, हाइड्रोजन बोन्डिङ प्याटर्नहरू के हुनसक्छ, राइट इन्टरमोलिकुलर कन्ट्याक्ट्सहरू कहाँ छ भनेर वी क्यान डु अल दोज काइन्ड्स अफ प्रोटिन स्ट्रक्चर प्रोपर्टी एनालिसिस। यसको इलेक्ट्रोस्ट्याटिक फिल्ड पोटेन्सियलहरू प्रोटिनको सराउन्डिङमा कस्तो छ भनेर, एमिनो एसिड्सहरू, यसको पिके भ्याल्युहरू, प्रोटिन सोल्भेसन इनर्जिजहरू र डिफ्रेन्ट इनिभिटिङ फ्याक्टर्सहरूसँग वा एक्टिभेटर्सहरूसँग बाइन्डिङ कोफिसियन्ट के छ, बाइन्डिङ कन्स्ट्यान्ट के छ भनेर वी क्यान कम अप विथ अदर काइन्ड्स अफ एनालिसिस। एन्ड फेरि म के भनिरहेको छु भन्दाखेरि अल दिस इज भेरी डिफिकल्ट टु डु म्यानुअली। यो म्यानुअली गर्न धेरै गाह्रो छ। सो दिस क्यान बी डन इफेक्टिभली विथ द हेल्प अफ कम्प्युटर साइन्स। कम्प्युटरबाट चाहिँ यसलाई चाहिँ एकदमै गर्नको लागि चाहिँ एकदमै सजिलो छ, बिकज यसमा के छ भन्दाखेरि देयर आर मेनी मेनी डिफ्रेन्ट कम्पोनेन्ट्स। So these tools it will help to explain evaluating both the experimental and theoretical structural models, right? यो एक्सपेरिमेन्टबाट आएको र चाहिँ हाम्रो theoretically बनेको structural modelsहरूलाई चाहिँ इभ्यालुएट गर्न चाहिँ it will help. So similarly अरू पनि छ, right? अरू डोमेन्सहरू पनि छ। So you can do biochemical simulationsहरू पनि गर्न सकिन्छ। So dynamic systems modeling to simulate the chemical reaction involved in the metabolism. So simulation can extend from individual metabolic pathway to transmembrane transport processes and even properties of the whole cell or tissue. So this is simulation चाहिँ अब अझै it will be extremely complicated है त, एकदमै complicated पार्टहरू हो। कसरी इन्डिभिजुअल इन्टरेक्सन, अब यो चाहिँ मोर अर लेस होलिस्टिक एप्रोच हामी आइडेन्टिफाई गर्न खोजिरहेको छौँ, राइट? सो नट अबाउट द इन्डिभिजुअल कम्पोनेन्ट कसरी इन्ट्रेक्ट गरिरहेछ भन्ने होइन कि ओभरअल कम्पोनेन्टहरू सेलमा भएको डिफ्रेन्ट कम्पोनेन्ट, ए कम्पोनेन्ट, राइट, बी कम्पोनेन्ट, सी, डी, ई, एफ, जी, एच, सबै कम्पोनेन्टहरू हाउ दे आर इन्टरेक्टिङ विथ वान एनदर। सो त्यो इन्टरेक्सन गर्नको लागि फर्स्ट चाहिँ वाट यु निड टु डु इज अ computationally you can do it, right? It is computationally intensive, but if you have sufficient computational power, then यिनीहरूको सबैको चाहिँ प्यारामिटर्सहरू चाहिँ स्टडी गरेर के चाहिँ हुन्छ त्यसको प्रपर्टिजहरू एन्ड हाउ इट विल इन्टरेक्ट विथ अदर सबै यिनीहरूको प्रपर्टीहरू चाहिँ आइडेन्टिफाई गरेर यु क्यान बिल्ड दिस काइन्ड्स अफ बायोकेमिकल सिमुलेसन्सहरू। So biochemical and cellular simulation traditionally have relied on the ability of the scientists to describe a system mathematically, right? You more computational mathematics, computational biology developing a system of differential equations that represent the different chemical reactions and fluxes occurring in the system. So Isma, what you do is that you generate different differential equations, right? So differential equations you can solve it. And then about the differential equations or the solutions, they can be extremely complicated, and then you can come up with certain kind of models. So the differential equations or have you studied differential equations?
Sir, math ma?
Math differential equations
sir.
The differential equations they are quite complicated, right? So differential equation solutions unique solution no saksa, right? Differential equation function नै हुन सक्छ solution. So what you can do is that because there are many interacting parts and you have many differential equations, right? So the differential equations धेरै कुरामा depend गर्न सक्छ। Now if you have a cell, right? Cell you want to model certain concentration of any reagent, ion concentration you want to model. So you have to identify all the processes that's occurring in the cell, right? यो भित्र आइरहेको छ, कुन प्रोसेसबाट भित्र आइरहेको छ, at what rate, कुन रेटमा आइरहेको छ, right? At what rate it is going out, diffusion अब कुन रेटमा यो बाहिर अर्को कुनैबाट चाहिँ कुन रेटमा आइरहेको छ, भित्र आइरहेको छ, राइट। सिमिलरली एनदर कुन रेटमा फेरि अर्कोबाट भित्र आइरहेको छ र यसले यसको रेटले यसलाई इम्प्याक्ट गर्छ कि गर्दैन, राइट। सो यसको अपटेक जुन छ, यसको अपटेकले यसलाई इम्प्याक्ट पार्छ कि पार्दैन, यसको प्लस हुन्छ कि माइनस हुन्छ, राइट। अल दिस थिङ्स यो एडिटिभ हुँदै जान्छ एन्ड यु हेभ टु डेभलप डिफ्रेन्ट काइन्ड अफ डिफ्रेन्ट डिफरेन्सियल इक्वेसन्सहरू जेनेरेट गर्नुपर्छ। सो नाउ इट विल बी एक्सट्रिमली कम्प्लिकेटेड है त। यु हेभ टु डिफ्रेन्ट रियाक्सन्सहरू कुन कुन छ, रियाक्सन्सहरू फ्लक्सेसहरू के छ, सो इनसाइड आइरहेको छ, आउटसाइड आइरहेको छ, अल यु हेभ टु डु द क्यालकुलेसन्स। तर मैले भनिसकेको छु है, फर्स्टली हुन्छ नि, यो भनेको एउटा टुल हो, दिस इज टुल है त। टुल हो, टुलको मिनिङ के हुन्छ भन्दाखेरि सुरुमा यु हेभ टु डु द एक्सपेरिमेन्ट्स, एक्सपेरिमेन्ट्स गरेपछि मात्र यु क्यान युज दिस टुल्स। एक्सपेरिमेन्ट गरेको छैन, इफ यु डोन्ट नो देयर इज दिस प्रोटिन, यो प्रोटिन छ, यो प्रोटिनले चाहिँ सोडियम आयन्सहरूको अपटेक गर्छ, एन्ड देयर इज अनदर प्रोटिन, राइट, एन्ड द्याट प्रोटिन विल to that protein the sodium ion can come out of the cell. So that information you have to have in the beginning, कुन रेटमा गर्छ, राइट, एट वाट फ्याक्टर्स, सबै थाहा हुनुपर्छ। एन्ड थाहा भएपछि मात्र त्यसलाई चाहिँ इनपुट गर्ने, र इनपुट गरेपछि त्यसपछि त्यसलाई स्टडी गर्ने, राइट। सो दिस इज अल अबाउट द बायोकेमिकल सिमुलेसन। So however, new software tools can build the mathematical framework of the simulation automatically from the description provided interactively by the users, making mathematical modeling accessible to any biologist who knows enough about a system to describe it according to the conventions of the dynamic systems modeling. So अब अहिले चाहिँ के छ भन्दाखेरि देयर आर सर्टेन टुल्सहरू छ, राइट। सर्टेन टुल्सहरूले चाहिँ नाउ यु डोन्ट ह्याभ टु डु द म्याथमेटिकल कोडिङ एज, त्यस्तो केही गर्नु पर्दैन, यु जस्ट ह्याभ टु। अब यो चाहिँ यो केमिकल इन्जिनियरिङहरूबाट चाहिँ आएको सफ्टवेयरहरू हुन्छ, यु जस्ट ह्याभ टु स्पेसिफाई है त। यो चाहिँ यु क्यान कन्सिडर अ सेल है त, यो होल सेललाई चाहिँ एउटा रियाक्टर जस्तो यु क्यान कन्सिडर। सो कुन रेटमा चाहिँ आइरहेको छ भनेर यु जस्ट ह्याभ टु प्रोभाइड द्याट नम्बर, अनि त्यसले चाहिँ यो सबै कम्प्लिकेटेड डाइनामिक सोल्युसन चाहिँ आफै गरेर चाहिँ यु क्यान प्रोभाइड यु विथ द्याट एन्सर्स। सो द्याट इज अल अबाउट द बायोकेमिकल सिमुलेसन चाहिँ, एकदमै कम्प्लिकेटेड हुन्छ, बट एउटा चाहिँ जस्ट यु हेभ टु अन्डरस्ट्यान्ड दिस अ होलिस्टिक एप्रोच अफ अन्डरस्ट्यान्डिङ द लिभिङ सिस्टम। सो नेक्स्ट इज द होल जिनोम एनालिसिस। कम्प्लिट जिनोमलाई पनि एनालिसिस गर्न सकिन्छ, राइट। सो एज मोर एन्ड मोर जिनोम्स आर सिक्वेन्स्ड कम्प्लिटली, द एनालिसिस अफ रॉ genome data has become more of an important task. So whole genome analysis project, now we have this next generation sequencing bhanja, NGS. So your next generation sequencing project, we have lots and lots of data. So your data like what to do with that data. So what you can do is that you can what you can do is that you can do the annotations. Annotation गर्न सकिन्छ, यसमा you can do the यसमा gene finding गर्न सकिन्छ, you can find genes at the gene promoter regionsहरू कहाँ छ भनेर you can identify at all those sequences, the homology informationsहरू, so all that kind of यसमा चाहिँ के गर्न सकिन्छ त यो जिनोममा। So all that kind of information you can compare this genome with other genomes है त। So you have the genome, and then you have this human genome. Now what you can do is that human genome, what you can do is that you can compare these genomes, right? You can compare these genomes. Similarly, you can compare human genome or the chimps genome with the mouse genome, right? What are the similarity, what are the dissimilarity? So all these kinds of analysis, it falls under the whole genome analysis, and for that there are different tools, right? So NCBI and other organizations are making a concerted effort to provide useful web interfaces to genome data so that the users can start from a high level map and navigate to the location of the specific gene sequence. So हामी के गर्न सक्छौं, we can input our genome into these web services, and यहाँबाट के गर्न सकिन्छ भन्दाखेरि you can navigate लोकेशन्सहरु, यो लोकेशन्सहरुमा स्पेसिफिक जिन लोकेशन्समा you can navigate, and genome investigation is far from the only issue in genomic sequence analysis, however, annotation framework, you have to annotate the genome. So if you sequence the genome of a cow, cowको genome sequence भयो, यो cowको genome sequence भएपछि, so what are the other things that you have to do? You have to identify how many genes are there. So you have to identify other many regions. यो जिसमा कहाँ छ junk DNA कहाँ छ, how much is the junk DNA, all that things, all that kind of information, annotations गर्ने पार्टहरु, right? त्यसको लागि फ्रेमवर्कहरु हुन्छ, कुन फ्रेमवर्कमा जाने, which integrates genome sequence with the results of gene finding analysis and sequence homology information. So यसको चाहिँ फर्स्ट यो साइटमा चाहिँ अब हेक्सोकाइनेजको जिन रहेछ, यसमा चाहिँ अर्को एन्जाइम रहेछ, यसो चाहिँ दिस इज एनदर साइट, यो अर्को साइट हो भनेर चाहिँ on the basis of homology information, sequence homology, humans बाट चाहिँ if you have already identified that in a humans, you can extract those information and are becoming more common, and the challenge of making and analyzing complete pairwise comparisons is also beginning to be addressed. So यो चाहिँ भर्खर हुँदैछ है त, यो कम्प्लिट जिनोम पेयरवाइज एनालिसिस भन्छ, जुन चिम्सको र ह्युमनको एनालिसिस गर्नको लागि चाहिँ स्टिल computers they find it extremely difficult, given that the genomes they are very large, and even to do a pairwise comparison between two genomes, it is computationally extremely challenging है त. यो म एउटा वर्ड प्रयोग गर्छु है त, यो computationally challenging भन्छु है त. एकदम प्रोसेसिङ पावर एकदमै हेभी लाग्छ, even we don't have that kind of processing power है त. So we'll continue, and even you can do primer designing है त, primer designing को पार्टहरु अब, so you can ask what are the physiochemical properties of that primer. So will they self hybridize or dimerize or not? So they should not have multiple targets within the sequence under the investigations. So all these kinds of things that we need to consider during primer designing. So that you can do with the help of bioinformatics. So what will be its annealing temperature, all that things that you can do with the help of this primer designing. So proper primer design is critical for the success of polymerase chain reaction, oligohybridization, DNA sequencing, and microarray experiments. So all these experiments, they require that you design the primer in the correct way. So primer must hybridize with the target DNA to provide a clear answer to the question being asked, but they must also have the appropriate physiochemical properties. They must not self-hybridize or dimerize, and should not have multiple targets within the sequence under the investigation. So how can you, so these primers, they should not bind to multiple sites in the target DNA. So if that happens, then you have the impurity. So that shouldn't be done. So for that, you need to design your primers, and there are many web-based services that allows user to submit a DNA sequence and automatically detect appropriate primers or compute the properties of the desired primer sequence, desired DNA sequence. So even you can do primer designing with the help of computer. So human like गर्न एकदम अप्ठ्यारो हुन्छ, even primer designing. So and then what you can do is that DNA microarray analysis. So यसमा पनि, so DNA micro मा चाहिँ this is expression analysis हो, right? Large scale expression analysis भन्छ. यो large scale the high throughput technologies हो. So DNA micro array ले चाहिँ के गर्न सक्छ भन्दाखेरि, it's a classic hybridization probe experiment. So यसमा चाहिँ, so the experiments that are being done at a very miniaturized scale. So microarray experiments are amenable to computational analysis because of the uniform standardized nature of the results on a grid. Results a grid of equally sized spots is identifiable with a particular DNA sequence. So यसको पहिले नै इन्फर्मेसन्सहरु पहिले नै हामीसँग हुन्छ, यो चाहिँ क्यारेक्टराइज हुन्छ एक्सट्रिमली, यो चाहिँ जुन ग्रिड छ यो डिएनए माइक्रोएरे को, दैट इज वेल क्यारेक्टराइज्ड, कुन सेगमेन्टमा चाहिँ कुन डिएनए प्रोब छ भनेर वी अलरेडी नो, एन्ड यसलाई चाहिँ हामी इमेज एनालिसिस गर्न सक्छौं, वी डु द इमेज एनालिसिस. अनि कुन रिजनमा कुन कलर आइरहेको छ भनेर वी क्यान डु यसको इमेज एनालिसिस गर्न सकिन्छ एन्ड क्विकली मान्छेले गर्दाखेरि इट विल बी एकदमै एगेन भनिहालेँ इट विल बी एक्सट्रिमली टाइम कन्ज्युमिङ है त एकदमै माइक्रोस्कोपिक हुन्छ सो कम्प्युटेसनल टुल्स आर रिक्वायर्ड टु एनालाइज द लार्ज माइक्रो एरे because the resulting image is so visually complex that comparison by hand is no longer feasible. So microscopic huncha yo ekdamai dot haru नै microscopic huncha and that dot so you this falls under the image analysis. सो कुन रङ छ यसमा है त यो प्रोब बाइन्ड भएको छ कि छैन राइट कुन प्रोटिन त्यो कम्प्लिमेन्ट्री डिएनए बाइन्ड हुँदाखेरि कुन कलर आउँछ राइट कति लेभलको आएको छ इन्टेन्सिटी सो द्याट ओन्ली अ कम्प्युटर क्यान डु सो द मेन टास्क द माइक्रो एरे एनालिसिस इज द that is currently done are an image analysis step in which individual spot on a micro area are identified. So your particular spots, that particular spot will identify and the signal intensity is quantified. So what is the intensity? We will quantify and clustering also. We will clustering step in which each spot signal intensities are identified. So you can we have to do this clustering experiments गर्नुपर्छ कुन रिजनको टिस्युहरूबाट चाहिँ यो आइरहेको छ सो यो कुनबाट आइरहेको छ भनेर विल डु द क्लस्टरिङ पनि गर्नुपर्ने हुन्छ सो यसको लागि चाहिँ विल डु सर्टेन काइन्ड अफ क्लस्टरिङ अल्गोरिदमहरू भन्छ फाइलोजेनेटिक एनालिसिसमा पनि पढ्छौँ क्लस्टरिङहरू गर्नुपर्छ so each is a clustering spot in which spot with similar signal intensities are identified कुन ले चाहिँ सिमिलर दिइरहेको छ भनेर यहाँनिर है त सो स्याम्पल 1 बाट आरएनए आइरहेको छ स्याम्पल 2 बाट आइरहेको छ सो अब कुन रिजन्सहरुबाट चाहिँ आएकोले चाहिँ सिमिलर दिइरहेको छ भनेर वी हैव टु डु द क्लस्टरिङ सो कम्प्युटेशनल सपोर्ट इज अल्सो रिक्वायर्ड फर चिप डिजाइन फेज पहिले यो चिप डिजाइन गर्दाखेरि पनि यु नीड द पावर अफ कम्प्युटेशनल टुल of the microarray experiment to identify appropriate oligonucleotide probe sequences for a particular set of gene and to maintain a record of the identity of each spot in the grid that may contain thousands of experiments. So how to design the probe, correct probe kasi design garne so that it will hybridize correctly and even that you have to need the power of computer. र यसलाई स्टोरेज गर्नको लागि राइट डिएनए माइक्रो एरेको स्टोरेज गर्नको लागि पनि यु निड द पावर अफ कम्प्युटर डेटाबेसेसहरू चाहियो सो इभन इन टु एनालाइज द DNA microarray experiments, you need to require the computational approach. So finally, we have the proteomics approach. The proteomics analysis garnako लागि पनि you need. So what you need to do is that proteomics is the study of proteome, the whole protein that is being synthesized. So before they are crystallized and biochemically characterized, proteins are often studied on a combination of gel electrophoresis, partial sequencing, and mass spectroscopy. यिनीहरूलाई पनि के गरिन्छ भन्दाखेरि इभन यी सबै इन्फर्मेसन्सहरूलाई जेल इलेक्ट्रोफोरेसिसको 2D जेल इलेक्ट्रोफोरेसिसको इन्फर्मेसन पार्शियल सिक्वेन्सिङको इन्फर्मेसन र मास स्पेक्ट्रोस्कोपी इन्फर्मेसनलाई पनि वी इन्टरप्रेट इन्टु अ डेटाबेस सजिलो होस् भनेर So 2D gel electrophoresis can separate a mixture of thousands of proteins into distinct components. The individual spots of the material can be blotted or even cut out from the gel and analyzed. So simple computational tool can provide some information to aid in the process of analyzing the protein mixtures. We can identify different molecular weight, isoelectric point of the protein sequence, right? All these things we can identify. And then it is also possible to compute from the protein sequence the peptide fragment that is created. अब यसलाई चाहिँ यो एमएस स्पेक्ट्रोस्कोपी गर्दाखेरि कुन फ्रेगमेन्ट जेनेरेट भएको छ भनेर इभन वी क्यान डु द्याट एन्ड द प्रोटिन्स is broken down by fragments by enzyme with a specific with a specific protein cleavage sites. All that kind of information what we can do is that databases generate गर्न सकिन्छ with the proteomics analysis को लागि. So certain protein को लागि कहाँ cleavage sites छ right यसको चाहिँ एमएस मास स्पेक्ट्रोस्कोपी गर्दाखेरि पेप्टाइड फ्रेगमेन्ट्सहरु के जेनेरेट भएको थियो 2D जेल इलेक्ट्रोफोरेसिसमा त्यो किसिमको सेल टाइपले कस्तो किसिमको 2D जेल इलेक्ट्रोफोरेसिस दिन्छ all that things can be analyzed है त सो यसको फिंगरप्रिन्ट्सहरु जेनेरेट गर्न सकिन्छ मास स्पेक्ट्रो एनालिसिसबाट सो दिस विल हेल्प है त कुनै नयाँ अर्गानिजम्समा इफ यु आर स्टडीing भने इट क्यान हेल्प यु र अदर आफ्नै कुनै रिसर्च क्यारी आउट गरिरहेको छ भने पनि सो विथ द हेल्प अफ दिस डेटाबेस इन्फर्मेसन यो डेटाबेसबाट इन्फर्मेसन गरेर यु क्यान क्यारी आउट रिसर्च quickly, quickly and efficiently. Right? So this is what we learn in our computational approaches to biological problems. So I hope you are convinced, right? So there is lots and lots to learn, right? From the computer group product. There is lots and lots of things to learn. We have identified in our class. So in our next class, we'll study about the history of bioinformatics. It's a history ma भनेर पढ्छौँ.